Hi, everybody. My name is Rich Hatfield. I'm a senior conservation biologist for the Xerces Society. I direct our bumblebee conservation programs, and for the last 4 or 5 years have been running the Pacific Northwest Bumblebee Atlas Project. I'm also helping to coordinate Atlas projects in a number of other states, including in the Great Plains, North Dakota, South Dakota, Kansas, and Nebraska, as well as Missouri, and then helping to coordinate the Minnesota Bumblebee Atlas Project as well. And then I'll just briefly let Leif introduce himself to you as well.
Hi, everybody. I'm Leif Richardson. I'm a conservation biologist with the Xerces Society. I've been with Xerces just over a year, and I am based in Southern California, where I lead the California Bumblebee Atlas. Before coming to Xerces, I did a lot of other work on bees, especially bumblebees, working as a consultant in academia and in other forums. And so I'm really glad to be with you tonight.
Great. Leif and I are hoping that tonight is going to be kind of fun. We both do a lot of these things and have heard more and more requests for more ID material. So instead of trying to pack it into a night where we talk about the Atlas and all these other things, we thought we would just do a standalone identification workshop. Takes things a little bit deeper and goes a little bit further than we do in those classes, potentially because we'll have a little bit longer attention span from all of you and from all of us, or from both of us who obviously after talking for 3 or 4 hours, we just get fatigued. So hopefully we can come in fresh, have some fun with this. There's some quizzes that we've made for you, as well as some quizzes we've made for each other if we get there. So I think we should have a good time. And thanks for being here. It's a real pleasure to be able to share this material with you, and it's nice to see so many people that are interested. As we present tonight, you're all probably familiar with Zoom at this point, but there's 2 features down in the boxes at the bottom in your taskbar that you should see. And we're going to use the Q&A down there for any questions that you have. So if you have, as we're presenting, we'll try to stop fairly frequently to address any questions that maybe have come up. And if you have questions, put them in there and we can address them at those breaks. And then if you want to chat with each other or connect with Leif or I, you can do that in the chat, and that's the place for that. Would also just ask that you please not use the raise hand feature, at least while we're presenting. It just, at least on my screen, creates like a bright flash, which I just find distracting. So if you could try to avoid using the raise hand feature, unless you want to taunt me, that would be appreciated. I think that should probably handle our sort of brief Zoom tutorial here. Hopefully you probably didn't even need that if you've made it this far. And I am going to go ahead and start sharing my screen, and we will get started.
The aforementioned conversation with children.
Yes, exactly. My son is downstairs and having a great time, and I just asked him to be a little bit quiet. So, Leif, would you just confirm for me that you can see my screen? Okay, great. Here we go. So Leif and I are going to talk to you today about how to identify bumblebees in Western North America. And this is not going to be a sort of technical how to use a key presentation. More or less, it's going to be a field guide to the bumblebees of Western North America. We're going to take you through each individual species that you could find in Oregon, Washington, Idaho, or California. And then we're going to go through some species groups that we consider to be difficult species groups, talk about how we discern between those individual species. And then we'll talk about males and how to identify males and take you through some of the males as well. And then, as I mentioned at the end, we've got a quiz to see how well you all learned our material, or probably better, how well we presented it. And then Leif and I are also going to quiz each other if we get there and have the time. So sit back, relax, and enjoy the show. Leif and I both work for the Xerces Society. We're a nonprofit based in Portland, Oregon. We have offices all over North America. We're nearing 75 different staff members working on a host of different conservation programs. We have a native pollinator program that works largely with farmers and working lands to try to create habitat for pollinators and other beneficial insects. Leif and I both work for the Endangered Species Program, which works on imperiled animals and either does direct science for them or sometimes uses community science like these programs to gather information about these imperiled species. And sometimes we're also advocating for those animals at the local, federal, and state levels. We also have an aquatic invertebrate program, including a large freshwater mussel program, and then a butterfly conservation program that works on Western monarchs and a whole host of other taxa, largely throughout the Western United States, but really throughout North America. And then also have a pesticide program that works as sort of an umbrella for all of these. As you can probably imagine, pesticides are a driving issue in invertebrate conservation. And so our pesticide program does a lot of work to help with the conservation of all of these animals. And as I mentioned, our on-the-ground conservation work through our pollinator program has actively, over the last 20 plus years, been putting habitat in the ground. And directly, we're nearing a million acres that we've supported pollinator habitat restoration on. And we've protected and restored over 1.5 million acres for rare and at-risk invertebrates throughout North America. So really a broad-reaching conservation effort. And we're also just celebrating our 50th anniversary. So Xerces turned 50 years not too long ago. And we were founded in 1971 by Bob Pyle, who's a long-term conservationist and butterfly taxonomist. And so over the last 50 years, we've grown dramatically to one individual making a difference to 75 plus individuals making a difference on over 1 million acres in North America. And as I mentioned, we use direct science and community science to gather information about imperiled invertebrates. You're all here likely because of our Bumble Bee Atlas projects. As I mentioned in the top of the hour here, that we're here talking about the Pacific Northwest and California Bumble Bee Atlas projects, but we're also running Atlas projects with a number of different partners throughout North America, and these programs are expanding. So it's a great way to gather information and have not just the 75 folks at Xerces contribute to conservation science, but also have thousands of volunteers all over the country, directly contributing to conservation. So thank you for all of you that have contributed to our Atlas projects, or if you're just interested for the very first time, welcome. If you're in a state that doesn't have an Atlas project yet, hopefully we'll be coming to a neighborhood near you soon. And if you're just joining us from somewhere random, you're welcome to. Glad you're here. We are a member-supported organization, and again, thank you to any of you that already are Xerces Society members. If you're interested in becoming a member, you can visit the URL that you see here on the screen, xerces.org/donate. All right. We've done our sort of introduction to Xerces Society, and now we're going to jump into why we're really here. And this slide says, how do we identify things? And sort of traditionally, if you went back, or if I went back probably longer than I want to admit, and when I first got into graduate school and was starting to study the bumblebees of California, I was sort of thrust with this guide that was written by my mentor and hero, Robin Thorpe, in 1983. And this was the guidebook to the bumblebees of California. And I was sort of thrust into finding animals and having to more or less put them on pins to kill them and put them on pins, and then to run them through these keys to try to identify them. So this is called the Dichotomous Key, and it's in a lot of areas, or a lot of books that we use to identify insects or almost any taxa out there use these dichotomous keys, where you go to sort of step one and you're presented with two choices, thus the name dichotomous. So two choices at each one. So if you look here on the left in the first sort of what we call couplet of Robin's key here to bumblebees of California, the first question is it a female or is it a male? And then you follow the number to the next couplet. So if we had female, we would go to step 2 here. If we had a male, we would go to step 25. And then we look at, you can see that the second couplet here is Malor space, longer than wide. Malor space as long or shorter than wide. And so you're presented with these series of questions, and ideally you follow these series of questions through all of the different couplets that take you to the series of questions that you get. And ideally you get a species name that matches the phenotype of the individual that you have. We are, these dichotomous keys are still incredibly useful for identifying organisms. And of course, we still use them. But we've also come a long way in the last 30 plus years in the development of digital photography and the availability of us to look at thousands of or tens of thousands of images and to be able to analyze those and look at them and maybe think about identification a little bit differently. And so especially in these atlas projects where we're actually using photographic identification techniques, we can't often run these animals through these dichotomous keys. We have to use a different approach. And so what Leif and I are going to take you through is sort of our approach of trying to identify these animals to help you become better. And you'll eventually develop your own techniques as well. But there's no harm in going back and looking at these old dichotomous keys. They're great resources. Robin has one for California. There are other resources that we'll share with you towards the end of the program, where we talk about all the different identification resources that are out there. So for tonight, we're gonna essentially use this form of a dichotomous key that if you've taken our Bumble Bee Atlas projects or classes or workshops before, you will have seen a diagram like this or one very similar to it. And we're more or less gonna assume that you know how to do this, that you can follow this dichotomous key from the insect that you're seeing on a flower, and you can get all the way to the bottom more or less without any help. So you can know that you have a bumblebee, you can know that you have a bee, you can know that you have a bumblebee, you can tell whether that bumblebee is a male or a female, and then you can also begin to tell, or you can more or less tell whether this is a Cytherus or a cuckoo bumblebee, or a non-Cytherus or a true bumblebee, a pollen collecting bumblebee. And the same with females. You can tell a cuckoo female from a non-cuckoo female. For those of you that might need a quick refresher on that last piece of whether we can tell a cuckoo male from a non-cuckoo male and same in females, we'll just do a quick refresher here. So here we are at that chart on the left. You'll see at the top, we have a male Cytherus versus a male non-Cytherus. Taking a quick look at their hind leg. Because cuckoo bumblebees are not pollen collecting animals, as you'll remember from our other workshops, they have sort of lost the morphological features that bumblebees have to collect pollen around the landscape. And so their hind legs, whether they're male or female, tend to look a little bit different. It's important to remember that with 50 plus species in North America and 250 plus species of bumblebees in the world, that there are differences here. You're going to see some variation in legs and the number of hairs and things that they have. But generally speaking, when we look at the hind leg of a cuckoo bumblebee, you will see that leg is convex. So this more or less looks like it's rounded. It's not a flat surface here. So this is actually rounded. And the surface is universally hairy. You can see pock marks everywhere throughout this feature, okay? And this is indicative that we have a cuckoo bumblebee. It's not perfect. There are better ways to do this by looking at genitalia and other things like that. But when we're looking at photos, it's hard to pull genitalia. So looking at this hind leg and looking at the degree of hairiness here is one of the main things that we're going to be looking at. And if I can compare that to a true bumblebee, so this is a pollen collecting bumblebee, and even though this is a male that actually doesn't collect pollen itself, it still has a morphological feature that looks more like a corbicula or looks more like a pollen-carrying feature than we saw on the cuckoo bumblebee. So you can see this is more of a flat surface here, and there's a shiny hairless area in the middle of this segment on the hind leg of the bumblebee. So I'll go back and show you the cuckoo. So again, we have this hairy area that's rounded, and in the true bumblebee, we have a hairless area that's shiny and flat. Okay, again, not a perfect character. It's not true with every species necessarily. You'll see some true bumblebees that will have more hairs than this, but this is a fairly reliable feature to look at to tell cuckoo males from non-cuckoo males. It is more reliable in the females, and so here we have the hind leg of a cuckoo female. Again, this is rounded, and while it is thick, it is not flat and triangular, and it is sort of convex coming out of the picture at you and has pock marks everywhere. So this is a hairy, fully hairy hairs coming out of in every direction that you see. And when I compare this to a true bumblebee, you can see we have a flat and even concave surface on this hind leg. It's surrounded by these long bristles that we call a corbicular fringe. And that corbicular fringe is used to hold pollen in place. And so these are for the pollen-carrying true bumblebees or the non-Siderus females. And of course, it could be possible that this feature would be obscured by pollen because it has been filled with pollen. But that makes your job even easier because any bumblebee that is carrying pollen is not a cuckoo bumblebee because cuckoo bumblebees do not collect pollen. So these are one of the major features that we're going to use to get to the bottom of that tree that I showed you a couple of minutes ago about whether we have a cuckoo bumblebee or a true bumblebee. And this is important because our cuckoo bumblebees, as you probably know from our other workshops, look a lot like our true bumblebees. And so knowing whether it's a cuckoo or a true bumblebee takes us to a very different part of the key or a very different part of the field guide that will give you and get you to an answer quicker, especially if you have a cuckoo bumblebee. Otherwise, you may find yourself getting confused. So remembering to come back to this decision tree and saying, do I have a male or a female? Do I have a cuckoo or a true bumblebee? Before I start trying to figure out what species it is really important. And then again, just to remind you, hopefully you've seen this material before. It's not the first time you've seen it, but just to remind you the morphology that we'll be talking about tonight, I'm just going to sort of run you through fairly briefly the different parts of the bee that we will be talking about and referring to throughout the program tonight to make sure that we're all more or less on the same page. So bumblebees, like insects, have three body parts: the head, thorax, and the abdomen. It is important to note that in some keys that you may find with bees, you may find the thorax referred to as the mesosoma and the abdomen referred to as the metasoma. And this is because thorax and abdomen actually have technical definitions in the entomological world. And bees actually have one segment of the thorax that's fused to the abdomen. So they're not, it's not a true thorax and it's not a true abdomen. So these body parts in sometimes in technical literature and key would be called the mesosoma and the metasoma. Generally, tonight, Leif and I will be using the term thorax and abdomen, but you may see those in technical keys. On the head, important body parts that we'll be paying attention to would be the vertex, which is the very top of the head, sort of behind these light gathering eyes on the insect called the ocelli. The colors of the hair on the vertex will be important, as will the color of the hairs on the face. And these are going to be down near the antennae. We will also be talking about the antennae some. So the antennae are also on the head. And then on the thorax, you will have the wings. Sometimes the darkness of wings can be an important character. And of course, the legs are also attached. And we already talked about why the legs are important for discerning male versus female and cuckoo versus true bumblebee. And then on the abdomen is broken up into tergites, 6 tergites in females, 7 tergites in males. And these are the segments. And we're going to be talking about the color of these different tergites and the pattern that we see on the abdomen overall. In addition to the tergites, there are also corresponding sternal segments that are on the underside of the abdomen, S1 through S6. Basically, if you imagine this being a 3-dimensional bumblebee instead of a cartoon, once we got to the side here, there would be sort of a juncture and you'd have a separate segment on the bottom side of the bee. And those are the sternal segments. So you will hear us referring to the sternal segments in addition to the tergal segments for some bees. Last thing here, just to remind you, I mentioned these 3 light gathering eyes on the top of the head. Those are called ocelli. And then we have the large compound eyes on the sides of the head. So that's a really quick overview of the bumblebee morphology. And then just getting a little bit closer as we look at the face or the head of the bee. Some of the things that we'll be talking about tonight will be the cheek or the malar space on the bee, where we're looking at the ratio of the length of this space to the width of this space, and this will be important. There are some species for which we look at the length of the different segments on the flagellomeres. So these segments of the antennae after the ball. So we have this long segment coming out of the face. There's kind of a ball joint here. And then these segments, 10 of which exist in females and 11 of which exist in males. Sometimes the ratio of the first flagellomere to the malar space matters. And sometimes we may be looking at the ratio of the first segment to the third segment and other things like that. You may see these in technical keys, and Leif and I may be referring to those somewhat tonight. And then just to give you another picture of what malar space looks like, these are 3 species in the Pyrobombus, all that have different length cheeks. On the left here, we have Bombus Flavifrons, which has a very long cheek. We have Bombus Kensis in the middle, which has sort of a medium long cheek. And then we have Bombus Mixtus on the far right here, which has sort of a medium short cheek. And so what we're really looking at is the distance between the bottom of the eye and the top of the mandibles. But it's not just that length that we're looking at. We're really paying attention of the ratio of the length of this space versus the width of the space. And it's hard to see in these straight on photos here, but there are actually 2 mandibular hinges that is probably best seen on this photo on the left. And we can't measure it here because the angle is wrong, but looking at the width of that versus this length and the ratio of those is very important. And we can see a better picture of that here. This is Bombus Aphenus, which is a Bombus sensu strictus species related to Bombus occidentalis, the western bumblebee. This species is found in the eastern United States. It's federally endangered and was listed in, I believe, 2017. But here you can see better that ratio. The length here is from the bottom of the eye to the top of the gap here in the mandibular. And then the width is actually from point to point in this mandibular hinge. And we're interested here in, is this wider than it is long, or is it longer than it is wide? Here in this case with Bombus Aphenus, this is a short-cheeked bee where the width of the mandibular hinges is a bigger number than the length here if I was to measure this, say, with a micrometer. So that's sort of a brief introduction to the characters that we'll be discussing tonight and the features that you'll be looking at as you start to think about identifying bumblebees. And I'm going to turn it over to Leif now, and we're going to do this a number of times tonight. And we've practiced this a little bit, but I'm going to try to give Leif control of my screen here so we don't have to switch back and forth. So Leif, I think that you should now have control of my mouse. And I'm going to stop talking and mute myself.
Great. Zoom confirms this, so let's hope they're right. Okay, great. Thanks, Rich. Oh, and so Rich, before I start, there was a question I thought we should we should answer live. Maybe you want to answer this. So this is from Ashley, who asked, I've been struggling with differentiating the different tergites of the abdomen. As you go through, would you mind pointing out how to know which tergite is which, other than counting from the thorax abdomen connection? Do you want to say something about that? And then we'll both focus on that as we go through.
Yeah, we can try to focus on this. I think this is, you're not alone in your struggle with this. Sometimes I look at photos and I struggle with this. And this is largely because bumblebees in particular are very hairy creatures, and some species have more and longer hairs than others. And those hairs often obscure the joints that we would normally see on some sort of other species of bees. And so some of this becomes a judgment call. Some of it becomes just having looked at tens of thousands of these individuals. And some of it is just, looking at the right place to find that juncture, or looking for transitions. I don't have a whole lot to say other than I think it's probably a good idea for you to, if you're really curious and want to take this deeper, is to either collect some specimens of yourself with the appropriate permits and everything that you would need to do that, or to get your hands on a teaching collection so you can actually look at this under a microscope or in a laboratory in person, so that you can do things like take a pin and sort of, pull those hairs apart a little bit and actually take a look at that juncture. Yeah, so I don't have anything other to say there, Leif. I don't know if you have anything to add, but yeah, I think that's a common struggle that people deal with in bumblebee identification.
Indeed. Yeah, I agree. A common problem. I think you handled it well, the answer that you gave. And I'll just add that sometimes it's useful to try to start counting from the junction that you mentioned between the thorax and the abdomen, because you can definitively tell which segment is number one if you can see it. And conversely, sometimes you start at the other end and you can definitely see 6. It's the pointy one with the sting sticking out of it, for example. But so there are some little tricks that you'll learn. But absolutely, what Rich says is right. You got to put in the work to start to learn this just based on examining live and pin specimens of bees. So great. We'll talk more about that over the next hour and more. So I'm now going to go through some of the basics of how we use color patterns to identify bumblebees to species. And as you heard, the technical way to identify bumblebees relies on a mixture of hair color patterns and other morphological characters. And we are not going to show you a dichotomous key. We're instead going to focus on the hair color patterns. And the reason we do that is actually that for this level of expertise or identification, there aren't very many morphological characteristics that you can use to distinguish one bumblebee from another. Another way to put this is if you were to take all of the hair off of a bee, and you could do this with a pin or something, I would probably not be able to identify any bee like that to species. They're exceedingly difficult to distinguish if they don't have the hair, with a few exceptions. So we use hair color. Unfortunately, it's really highly variable. And so that is a challenge that many of you are already familiar with. And let's see here. I'm now trying to advance the slide. Rich, I don't see it moving. Let's see here. Rich, can you hear me? Oh, okay. Okay, thanks. I was having trouble advancing slides. I may need you to do this.
Okay.
But,
sorry,
okay.
I forgot I was muted.
No, it's fine. So hair color patterns are often enough for ID, but there are going to be a lot of exceptions. And these 3 species that we show in cartoon form here give you a sense of how similar the gross color morphology can be. Rich, do you want to go to the next slide? All right, so these color patterns are variable between species, and unfortunately, they're highly variable among within a given species. So many of the species that occur in Western North America have a broad range of color patterns, some featuring bright red, some replacing the red with black, some replacing the yellow with white, and so forth. And so this image here shows you one species, Bombus Bifarius. Some taxonomists now believe this is 2 or more species. Others point to the idea that it's just one. I'm going to pretend it's just one for now because we haven't heard the last from them about this. But the point is that this is Bombus Bifarius, a species that is starkly different in different parts of its range. So if you're in California, you will only, with maybe the exception of the White Mountains in Eastern California, you will only find these black and yellow individuals. Whereas in a lot of the rest of the West, Oregon, Washington, you'll find this bee with some red on it. So Rich, do you want to advance again? So
arrow keys work on your keyboard. Just give that a try.
Yeah, they are not working.
Okay.
Great, thank you. So so we separate additionally, this variability can be changed through evolution on very short timescales. And so we see given bumblebees adapting over longer than our lifetimes, but relatively short evolutionary timescales to become similar to each other where they occur. So this second image here shows you complexes of species that look very similar when they occur together. And if you'll just go forward one more, these purple boxes are delineating some of the groups that occur in California and as well as in the other Western states. And so the point is that in any given area, we see multiple species converging on a similar color pattern. And we think that this is mimicry. It's mimicry where the color pattern sends a signal to potential enemies like predators and perhaps parasites or parasitoids that this potential food item could sting you. And so the more animals that look like this, the stronger the signal is that is sent to those predators. And so we think that that's why natural selection works to create these mimicry complexes. It's not good news for us as humans trying to identify the bees, but it's important for you to understand this so that if you travel, if you are unfamiliar with the bees in your area and you go outside and look, you'll start to see that there are multiple species in an area, but they can be very similar in their color patterns. Go ahead with the slide. So using color patterns, we're going to start with a kind of a very basic sort of thumbnail cartoon image of each bee. And here we're showing you a graphic from the California Bumblebee Atlas. This is female bumblebees found in California, and they are divided into 5 color pattern categories. Now this is for California. However, these are most of the species that occur throughout the West, with maybe 1 or 2 exceptions, or 3. And so this should be just fine outside of California also. But you can see that I've divided the bees into a category that has red or orange hair. Second, a category that has yellow and black striping, but the first abdominal segment, that T1, is going to be yellow. Then third, a group that is also yellow and black striped, but the first abdominal segment, T1, is black. Fourth, bees that contain or feature some amount of white hair somewhere on the body. And finally, the 3 cuckoo bumblebees. And go ahead with the slide, Rich. And so the idea of this field key, as I like to refer to this thing, is that it just gets you in the right room. You are looking at a bee, like the photo here. You don't know what it is, but obviously it's got prominent red bands on it. So you go to that number 1 part of this sheet, and if you peruse, you start to find a bee that closely resembles the one that you're holding. You can then go to field guides for more information and to really drill down on what the identification is. So a key like this isn't adequate all by itself, but it is where you can start if you're a community scientist trying to understand bumblebees better. Go ahead. So I'm now going to walk you through those groups backwards, quickly introduce them, and then I will present the first of them, which is the red. I'm going backwards just so that the last one you see is the red, and I will start talking about that one. So we will talk about those cuckoo bumblebees. We'll talk about the four species that occur here that can have white hair. Next, we'll get into those species that are striped and have T1 black. Next, we have the same except T1 is yellow. And finally, we'll get to those red or orange tails. So red or orange hair somewhere on the abdomen. And with that, we'll get into the individual species. And so first, we are going to talk about Bombus Hunty Eye or Hunt Bumblebee. This part of the talk will look at each slide will look like this, same format. And so what you're going to see in the bottom left-hand corner is obviously the range map. And you can see a thumbnail map that shows the full distribution in North America. And then the larger map is showing you all of the known observation records, or all of the observation records, specimen and observation records that we were able to pull together, divided into three categories according to how old or how recent they are. And so you can interpret the green as the historic condition, the yellow as the first decade of the 21st century, and the red as the last 10 years, the second decade of the 21st century. We're using these cartoon figures from Bumblebees in North America, the field guide, are slightly different than the ones you have been seeing, to show you the color pattern. And we're also going to use arrows, if you want to go forward, Rich, to point at things on each bee that show you the distinguishing marks. And so, and then of course we have some text at the top right, which tells you which species this can easily be confused with, and a short roundup of the characters that we're interested in here. So these slides are all going to show you female bees, workers, sometimes queens, and queens and workers are often quite similar, not always. We'll talk more about male bumblebees later tonight, but so first we'll just be talking about these females. So Hunt Bumblebee, this is a common and widespread species in Western North America. It's found in a range of habitats, and you can see that in the most recent time period, the last 10 years, there have been numerous observations around the West. And it's found in California, mainly in the montane parts in the Sierra Nevada and east of the Sierra Nevada. It's found broadly in Oregon and Washington, east of the Cascades, and broadly in Idaho as well. This is a bee that has a cheek, a malar space that is about square. So it's about as long as it is wide. Unfortunately, you won't be able to distinguish it based on being especially short or long. It has short, even hair. So it looks a little bit velvety. All the hair is about the same length, and it looks like the bee combed itself before leaving the nest. In fact, it may have with body parts that they groom themselves with. The corbicular fringe, so the hairs around the pollen carrying the pollen basket on the hind leg are black, and that's to be distinguished from orangey red hair, which we'll see on another. And then on the thorax, where the upper left-hand purple arrow is pointing, there is entirely yellow hair. There will be no black hair mixed in there, and that is distinctly different from many of the other bees that have red hair on them. Rich, you can go ahead to the next slide. And I'll say Hunty Eye, or Hunt Bumblebee, which we just covered, is one of the only ones where the color pattern is very straightforward. It doesn't vary very much across the range of the bee. And so here's one where we do see quite a bit of variation from place to place. And this will be more the rule than the exception, this variation that I talked about earlier. So here we have the black-tailed bumblebee, Bombus Melanopagus, or some people say Melanopidges. None of us are native Latin speakers, and so I don't think it matters how you pronounce these words. You can see that this is also a bee that's quite broadly distributed in the West, particularly along the West Coast. This is an abundant species. It's actually the first or second, well, let's say the second most common species in California, and I know it's quite common elsewhere. This is another one with a square cheek. This is one that it does not have super short hair, like Hunt's Bumblebee, which we just talked about. This one is a little bit more medium length, a little bit shaggy, just a little bit. Importantly, on the thorax, on the front part of the thorax, we have a mix of black and yellow hair in most specimens. You can sometimes see some of that in the back yellow segment of the thorax also, but look for that mix of black and yellow hair in the front band of the thorax, a prominent black band across the middle of it. And then in Most of the West, this bee will have these red stripes, these prominent yellow stripes, red stripes across T2 and T3. The one exception is California, where the bee is entirely going to be black and yellow, except for areas north of Eureka. So we're talking about a very short distance between Eureka and the Oregon border. You can start to pick up the red morphs, or a mix of characters between the two. So in California, this bee used to be referred to as Bombus Edwardsii. We now know that they're the same species, and really one of the only things that's differs in them is that color pattern. So it's just a few changes to a few genes. They look quite different, and were always thought to be different species until sometime in the last century when people figured this out. So Rich, you can go ahead. Next, we'll talk about Bombus Rufus Cinctus, or the red-belted bumblebee. This is a beautiful bee. It's very short hair, very neat, even hair, similar to the first one I showed you, Hunt Bumblebee. But this bee is not closely related to Hunt Bumblebee, and it actually has some characteristics that can help you to distinguish it from others. And so one of those is that it has a very short face. It has a very short malar space relative to its width. So check out that cheek, and you'll see just a little bit of space between the bottom of the eye and the top of the mandible. That, combined with short body hair and a very typical oval black hair area on the center top of the thorax, which can vary, but is almost always like that. And thank you, Rich. There's the short cheek. It's not the shortest. We will see other bumblebees with shorter, but this is what we mean by short. It's distinctively shorter than it is wide. Now, this bee had, unfortunately for us, Bombus Rufus Cinctus, or red-belted bumblebee, is probably the most variable of the bumblebees in North America in terms of its color pattern. It can have a lot of red. It can have red anywhere from T2 to all the way to T5, maybe even T6. It can have variable amounts of yellow. It can have black separate in there on parts of segments or on whole segments. One of the few characteristics that is almost always present is the yellow on T2 right in the middle. So if you look at those color pattern diagrams or cartoons, you see yellow in the center of T2 on almost every one of them. Just one exception, I think. This is a good thing to look for. So if you're starting to suspect you have red-belted bumblebee, well, it's not the only one that has yellow hair on T2 in the middle, but it should have it. If it doesn't, you should start thinking about other species that you may be looking at. This bee has an interesting distribution. It's found coast to coast, although not very far south. It's from the sort of temperate, northern temperate forest zone into the boreal zone and farther north across the east. And then in the west, it's in montane places and all sorts of other habitats. It is extremely variable, but that said, once you start to learn bumblebees and become conversant with them, this one is not as hard as it may sound to identify, despite all of its variation. So let's move on to the next, Rich. Thank you. Okay, so this is two-form bumblebee, Bombus bifarius. And so here's, this is the one that we talked about in that graphic where I showed numerous different color patterns on the same map. And I told you that this may be a bee with, that is 2 or even 3 species, we're not certain. It's very similar to a number of other bumblebees in the West. There's a distinctive characteristic you can use to identify it in many cases, and that is on the thorax. The purple arrow pointing down is pointing at black hair on the back part of the thorax, and it's medial, it's central, and it's surrounded by yellow hair. So there could be some admixture there of the two colors, but down the middle, you should see black hair pointing towards the abdomen. You can see that in the color pattern diagrams, where there's a V shape, a notch shaped black hair area that is pointing backwards. And we also often see black hair turn up on T2 in that same little spot where we saw yellow hair turn up on Rufus Cinctus, red-belted bumblebee. Well, here we have the black. In most of the West, this bee is going to have some amount of red hair on it. as you can see in this beautiful photograph. However, in California, we are going to expect to see both animals with red and animals with all black and yellow. So this can be pretty challenging, but I want you to remember that thorax characteristic. That's particularly important. Another good one is on the hind leg, the pollen basket has orange or orangey red hairs or bristles around the outside of it. Those are those bristles that are incorporated into the pollen mass for carrying home. They're structural, they hold it in place. Remember that we looked at some other species already that had black hair there. So that's a good one, a good characteristic for this species. And the last arrow that I haven't described is pointing at the mix of black and pale hair in the front part of the thorax. Not unique to this species, but it is present in almost every case on this species. So you can go ahead. Next, we'll talk about Bombus Silvicola. This is an interesting animal. It's a relative of most of the other ones that we've talked about in the red hair group. However, this one is found at mid to upper elevation across the West. We do not think it's of conservation concern, despite the relative paucity of records that you see on that map. Although we're still learning more about this species. But it is one of the ones that we think is restricted to some of the upper elevation habitats. And so it's beyond the scope of this talk, but you may have heard us talk in the past about threats to bumblebees. And one of those threats is climate change. And that is particularly a threat for bees that live in high elevation areas, such as this one. So that's something to keep in mind. Now, Silvicola is another one that has a square cheek, unfortunately not very distinctive or useful, except when comparing to those that don't have square cheeks. It has long, bushy hair, shaggy hair of varying lengths. That question about distinguishing the various segments of the abdomen, I think you can pretty well see them in this image, going from the first one being yellow, the second being red. Thank you, Rich. Yeah, you can kind of see the black integument or skin of the bee under the hair, and that's a good bet that you're seeing the break between the adjacent segments that actually overlap each other slightly. The one in front overlaps the one farther back just slightly. You won't always be able to see that black, but I wanted to point that out with this bee because with longer haired bees, we sometimes cannot see the segment breaks because the hair overlaps to the next segment. So it's a little place where you want to look extra carefully if you have a bee with long hair like this. So more about this bee, it has both black and yellow hair on the face and the vertex. That square cheek. It has yellow hair on T5. And let's see. And yeah, I think that's all I'll say about this one for now. We can go forward. And this is Bombus Flavifrons. So this is another one with bright red hair. As you can see, in this case, we have the red hair starting on T3, right? So we've got 2 yellows, then we've got 2 reds. This is a really striking and beautiful bee. It's just distinctively different in its color pattern in different places. This morph with these bright red color patterns is the one you'll see in most of the Pacific Northwest. Not the only one, but it's quite common. You'll see this elsewhere around the West. In California, we more often see a morph that has replaced the red hair with black hair. So you can kind of see that in those images that are grayed out, and we'll maybe talk about that a little bit later. This is another one with it's got medium length, somewhat shaggy hair. So if you compare back to your memory of the recent photos, this is not a short hair and this is not the longest hair either. It has a distinctively long cheek. So finally, here's a bee that has a longer than wide cheek area. And this one, I find it to be really not a question if you look carefully and you're looking at the right species. It has an awful lot of admixture of dark hair in the yellow areas of the front of the thorax, the back of the thorax. In some of the, it's even, it can be closer to blacker than yellow in some populations that lack the red. So in California, I think we're going to see both of these morphs, and I think that's the case in areas of the Pacific Northwest also, but look for this red one. And go ahead, Rich. All right, and then this is Bombus Centralis. This is one that has 2 yellow segments on the abdomen, then 2 red segments. This is a bee that has just a slightly longer than wide cheek. So it's not as long as the last one, as Flabberfrons, but it is distinctively longer than wide. And that should help to compare it to a number of the other species in this color pattern group who are part of the same subgenus of the genus Bombus, and they most of them have cheeks that are about square, as we've been saying. So look for medium long cheek, look for longer hair, not the longest, not like Silvicola, but longer, I would say somewhat like Flabberfrons, the last one we looked at. The thorax does not have black hair mixed with the yellow. So look at that, the area that Rich is pointing to there, the front of the thorax, it's all yellow. And then behind there on the back of the thorax, also all yellow. And you can see from our color pattern diagrams that this bee is another one that doesn't change very much across its range. This is a species that's found broadly in the West. We actually think it's doing fine, but we haven't done a thorough atlas in a lot of areas where it occurs, like California and Nevada and Utah. And so we are going to learn more about this bee as we move forward with atlas projects. And you can go ahead and go to the next. I think this is the last bee in this group, and this is Bombus Crotchii, or Crotch's bumblebee. This bee is really different from the others we've talked about a bit so far. So this bee is in a different group than the others we've talked about. It is very large, at least the queens are. It has very short hair, a very distinctively short cheek. If you get a good look at the face and you see that cheek, it is the shortest we've talked about yet. There is no mixture of yellow and black hair on the thorax. We've got this beautiful yellow segment and this black segment right next to it. And so that should distinguish it a bit from others that have reddish hair. And we do see red hair on this bee on the distal parts of the abdomen. So it can be segments 4, 5, and 6. And I want to say that you see that there are black color patterns also for this bee. In California, where this species is almost entirely endemic, we have, I think, we have 99% of the known records are in California. There are 15 from Baja California and a single one from Nevada. Otherwise, all of them are from California. So you won't expect to see this bee outside of the state of California in very many cases. About 75% of them have that red hair, but you should also expect to see it in the sort of all black and yellow pattern also. This is a species of greatest conservation need in the state of California. It's been petitioned for listing as endangered, and you can kind of get a sense of why if you look at that range map. The most recent time period there, the last 10 years, the large majority of records have come from south of the transverse ranges in places like Los Angeles and San Diego, where it can be abundant locally. And we are still seeing it farther north, but not too often. And the historic condition was that we had a lot of observations of this bee from the Central Valley and indeed up into Northern California. So go ahead. Oh, I was wrong with that. So this is Bombus mixtus, the fuzzy-horned bumblebee, another with red hair. And so in this species, we're looking at red hair towards the tip of the tail, like in Crotch's bumblebee, although this one is quite different in its aspect from Crotch's bumblebee. So this is one that has a cheek that's just a little bit shorter than it is wide. So this isn't square, it's small, it's shorter than it is wide. So that should be helpful in comparing it to some of its close relatives like Flabberfrons, the one with the long face that can look quite a bit like this, despite the red hair. So it has longish hair, as you can see, somewhat shaggy, a little bit uneven, a very strong admixture of yellow and black hair on the face, on the vertex, all over the abdomen, particularly in the front band of yellow and also in the back band of yellow in some cases. It is going to have variable yellow and black patterning on the first segments of the abdomen. So up to about segment 3 or 4, and then you will always have some red hair towards the tip of the tail there. Mixtus is a bee that we think is probably doing fine. It's secure. But we again have not done formal atlas projects in most Western states. And so we may learn more about this as we go along. In California, this bee occurs in montane areas as well as on the coast. And you can see that that is also the case in Washington and Oregon. So it's a funny set of habitats or ecosystems that this bee occurs in, both at sea level on the Pacific Ocean and quite high in elevation in the Sierra Nevadas, for example, and in the Cascades. We have other species that have a similar distribution. So there's something going on there where bumblebees know what they're doing. And those habitats may seem different, but maybe they aren't to the bumblebees. The last one, I think, in this group is frigid bumblebee. This one is, you can look at the global map there and see that this is a northern species, a boreal species of bumblebee that does occur in our area, but just sparingly in, I think it's only it's in the northern parts of Washington, Idaho, Montana. And then it does occur at higher elevation south through the Rockies at least as far as Colorado. And so this is another one that has red hair at the tip of the tail. It's in that same group of subgenus of the bumblebees. I haven't said the name yet. It's called Pyrobombus. Pyro, I think, meaning red. So many of them are red. This is one with a square cheek. It has long, shaggy hair. It has all yellow hair on the front of the thorax there with no admixture of dark hairs. The face is mostly black. The corbicular fringe, so the pollen basket bristles, are pale. And remember that we've looked at species with orangey red bristles. That would be, I think it was Bifarius. And then we've looked at those with black bristles. That's sort of most of them. And notice that there is no crescent shape on T2. T2 is all yellow. And I don't know this bee as well as Rich, who made this slide, and I believe that distinguishes it from Bombus mixtus, the last one that we looked at. So we won't see this bee in California. We don't expect to find it in Nevada, although who knows, at the highest elevations, it could be farther south, but do look for it if you're working in montane places farther north. And high country bumblebee is another one that usually has red hair. So this is a beautiful and not common species. You're lucky if you get to see it. This is another one that has a northern and montane distribution. If you look at that, the global range map, you can see that it is found as far north as Alaska and Yukon and Nunavut. It's found across Arctic Canada. And then it comes down through the Rocky Mountains as far south as Colorado, possibly New Mexico. There is a disjunct population in California, and there are several other mountain ranges, for example, in Idaho, where it's been found, and they are definitely disjunct from other populations. Despite Rich's hard work over the last 4+ years in the PNW, working on an atlas project with many of you, we have not recovered this bee in the mountain ranges of Oregon or Washington, with the exception of the northern Cascades. So this is one that we would have expected to find it, I think, at high elevation in Oregon, and it didn't turn up. So there are some questions about this bee. In California, it is mostly represented by these historic observations in the Sierra Nevadas. I believe there's at least one more recent one, but we really wonder what the status of this bee is like now, and going forward with climate warming, what's the future for these disjunct populations in isolated high elevation mountain ranges in the West. So this bee has a very long cheek. It's in a group of bumblebees that occur worldwide in Arctic and subarctic places, and it just happens to have a very big head, a very long face. You can't miss it, the shape of the face there. The hair is of medium length, definitely not short, sometimes somewhat shaggy. The thorax, where the hair is yellow, you're going to have no admixture of dark hairs. For example, that front band of the thorax is all yellow. The face is black. It's got black face hair, and then the vertex is black. I think you can sometimes get some yellow hair mixed in there also. This is a sensitive species in Washington and Oregon. I think at some point we'll get around to calling it that in California also, just in consideration of how limited its distribution is. All right, and with that, I am going to turn it over to Rich and ask first, are there questions that we might want to answer?
I don't think so. I think I've been trying to handle, there was a question to point out the tergites again, and also to point out the cartoon that we were referring to in the picture. And I've been trying to do that with the pen and other things. So I think we're good. I'm just gonna keep rolling. That's okay.
Okay, thanks.
Yeah, thank you. All right, now we're going to jump into the bumblebees, some of which will be the same species you've already seen, that have stripes that do not have red on their bodies, and that have yellow on the first tergal segment. And remember that the first tergal segment is closest to the thorax, the closest to the head. So jumping right in, the first species we're going to talk about is a species Leif introduced as Bombus Melanopagus, Bombus Melanopidges, the black-tailed bumblebee. Super common species, very early emerging species as well. In Oregon, Washington, and Idaho, we will see largely this species with red on it, although down near Ashland and south down to Eureka area is where we see this mixture, this transition from the red species in the north to the blacker species in California. And the black ones that we see here, like pictured on this slide here, have less admixture. So generally in the Pacific Northwest and in Idaho, we see a lot of black hairs mixed in on the face and in the front of the thorax, in front of the wings there. We don't see that so much or quite as much as we see in the Pacific Northwest, but you can still see there are black hairs intermixed in there. And in contrast to some of the other species we'll see, that is not the case. That is a good feature to be looking at. Another thing that we want to look at is the addition of yellow hairs here on T5. So this is T1 that we see that's yellow. You can see sort of a division between T2 and T3 here. So 2 here, 3 here, and then 4 is yellow, and then 5 is also almost always yellow, at least on the sides. And this is notch that we see here in the bumblebee in the cartoon is what gives this bumblebee its common name of the black-tailed bumblebee. So that is that species. Again, this species is very common, very broadly distributed in the West and throughout mountains, higher elevations in the Rockies as well. And this is another very, very common species and a very close lookalike to Bombus Melanopidges. This is the two-form bumblebee, Bombus Flavifrons. Leif also introduced you to the red color form of this as we see the further east we move, and sometimes at higher elevations in the Cascades as well. And there's also some red color forms actually in the San Juan Mountain or the San Juan Islands in Puget Sound. We see some individuals with red there as well. But largely in most areas, and especially in California, the species is going to have the color form that is exhibited here. And for identification features, a few things that we're going to look at here. Remember, this is that species that Leif mentioned, that notch behind the wings. That black triangle pointed backwards, best seen here in the cartoons, where it's not just a lack of hairs, it's actually black hairs that are pointing and go all the way back to the end of that thorax. It's a very clear picture that you'll see. This species also has some admixture of black and yellow hairs in front of the wings, but less behind the wings and almost none behind the wings except for that notch between the wings. And then, yeah, yellow here on the first tergal segment. Segments 2 and 3 are black. Segment 4 is yellow. And then you'll see segment 5 here behind that yellow stripe. This is segment 5, is all black. We don't see yellow on the sides as we did in Bombus Melanopidges. So those two species we just looked at, Melanopidges and Bifarius, are very close lookalikes. Another thing that we'll be looking at here are the red curvicular fringe and yellow in general legs that we see on Bombus Bifarius that we don't see as much in Bombus Melanopidges. This is Bombus Flavifrons, another species that Leif also introduced you to, generally as the red color form. But more commonly, especially in the West and at lower elevations, we see this species with black on it. There are, again, a population in the San Juan Islands that have red on it, and then sometimes at higher elevation. And as we move eastward into Idaho, we'll see more and more of the red color form. But certainly, like in Portland and Seattle and lower elevations in California and the West, we're going to see this species as it's pictured here. Again, very long cheek on this species, much longer than wide. This admixture of hairs is again very prominent in this species. Although it's important to note that behind the wings here, you can't see it very well in this picture, although it's pictured a little bit better in the cartoons here, there is still quite a bit of yellow hair, and that's going to differentiate it from another species that we're going to see later, Bombus Kensis. So just keep that in mind as we move forward, is that behind the wings there still is, well, I guess the important part is the amount of yellow hair is equal in front of the wings as it is behind the wings, which is not true in Bombus Kensis. And that's what we'll be looking for to differentiate those two species that we'll take a look later. And that one's in the white group, although we could have included it here. Bombus Scruticolos is another species that has the first tergal segment yellow with a stripe there. This species is somewhat common in our drier areas. I think of this as more of a lower elevation sagebrush steppe type habitats that we see in the Pacific Northwest, sometimes found as far west as the Willamette Valley, and actually quite a few observations in the Willamette Valley. So it seems to be okay with human habitation. We find it in urban and suburban areas at lower elevations. Really striking species and different than a lot of the pyrobombus that we've been looking at. You'll notice if you look at the photo, this has very short, even hair. The hair on the face is very black. The vertex as well on the top of the head, which we see here, is also quite black. The thorax is mostly yellow, although we see usually see a strong black spot in the middle of the thorax, right between the wings. And then oftentimes there's a crescent of either yellow or red or brown on the second tergal segment, and maybe best seen here in this cartoon version up here, where it's black on the sides and towards the back, but it's sort of brown in the center there. And what I often see in this species, at least in Oregon and Washington, is that the queens tend to have more of the yellow color form, where I see this more in the queens, and the workers tend to have the brown color form here in Oregon. It may be different in different regions, but that's been my personal experience with this species in the Pacific Northwest. Bombus pennsylvanicus, the American bumblebee, is another species with yellow on T1 and stripes on its abdomen. And this species largely absent from the northern portion of its range in the western United States. We have observations from Southern California that have come in recently, but really nothing north of the Los Angeles region there. So nothing in Northern California, nothing in Oregon. We did have one observation from the Pacific Northwest Bumble Bee Atlas in eastern Idaho from a couple of years ago. And there is an observation from southeastern Washington from a number of years ago, not from the Atlas, but from another researcher that has seen this species. This species has been petitioned for endangered species protection, and it's often referred to as Bombus sonoris in the western United States, especially down in the southern portion of its range. And this means Bombus sonoris generally means that it's going to have more yellow here on the first tergal segment, as well as behind the wings. In the eastern US, we'd more see this color pattern with more black on T1 and less yellow behind the wings. The species has a very long cheek, like Bombus Flavifrons, probably longer than Bombus Flavifrons. It has black on the face, black on the vertex, and also another character we pay attention to with this species is these very dark wings. You'll notice if you look at some of the other photos that we've been showing you, the photos are a lot clearer or more easy to see through. They're quite dark in Bombus pennsylvanicus, and that's a character we can use in photos and in the field. Bombus Navidensis, this is another sort of sagebrush steppe species that we find at lower elevations and in urban and suburban areas sometimes. This also has very short hair, just like the species we looked at a couple of slides ago, Bombus Scruticolos. This one is different than Bombus Scruticolos in that it has yellow on T1, T2, and T3 versus, remember, Scruticolos had yellow on T1 and just a crescent here on T2. This species has 3 yellow segments on its abdomen, but also shares that character of having a largely yellow thorax with a black spot in the middle of it. Can't see it in the photos here, but this species has a black face and a black vertex. I will also note that this character below the wings here, a little difficult to see in this photo, but it has yellow. Basically, if you draw a line straight down below the wings here, it is yellow in front of that and black behind it. And it is a very noticeable line when you see this species, in a photograph, the right photograph, or in person. That's another character that you can look for. This species appears to be quite stable. Has a similar distribution now than we've found historically. We contrast that with Bombus morrisoni, which is a very close look-alike species, Morrison's bumblebee. This is the species we're concerned about. It's a species of greatest conservation need in Oregon, Washington, Idaho, and California. Again, another sagebrush species with short, even hairs. This species is a beautiful, beautiful animal. It has the brightest color of yellow that I think I've ever seen on a bumblebee. Very striking creature to come across. It is one thing that I will say is that this species, while we haven't detected it very often in the last several years, this species thrives in many of the areas that are undersampled. So this would be a plug for you to head out to southeastern Oregon in the areas where this species does seem to still be extant, to try to see it in the wild, because it really is a beautiful animal and is worth trying to see. A few things we use to differentiate the species from its close lookalike of Bombus Navidensis. Instead of having black on the vertex, it has yellow on the top of its head or on its vertex, and there's no black spot in between the wings. Usually the entire thorax is this thick carpet of this golden yellow color. We also often look for this crescent of yellow on sometimes on the 3rd and sometimes as far down as the 4th tergal segment. So unlike Navidensis, which always has the first 3 segments that are going to be evenly colored yellow, we often see this crescent on the 3rd or the 4th on Bombus morrisoni. This species also has a very short cheek in contrast to Bombus navidensis. Navidensis has a very long cheek. Bombus morrisoni has a rather short cheek. Moving on to Bombus vagans. This is a species that again has like some of the species we looked at, has a cross-continental distribution. I would say that previous to the Pacific Northwest Bumblebee Atlas, we didn't recognize how far west this species has actually seems to be extant. So a lot of these records in the Eastern Sierra are brand new to our understanding of bumblebee distribution. So pretty neat. This is a medium to long cheeked bee. It tends to have rather long hair, a little bit shaggier than some of the other species, certainly than sort of Navidensis or morrisoni that we've just been looking at, or than Scruticolos, which could be confused with it. This species has all bright yellow hairs on its thorax. Similar to Navidensis, it's going to have a black spot, or usually have a black spot here in the middle. Usually mostly black face and vertex, although some yellow can creep in there. And here we're going to have yellow on the first 2 tergal segments, although the 3rd tergal segment is almost always black in this species. Occasionally, you will find a little bit of yellow on T5 on this species, although I would say that's a fairly rare character, at least in my experience with this species. And that ends the species that are striped with yellow on T1. I'm now going to pass it back to Leif, who's going to talk about species that are striped with black on T1. So go ahead.
Thank you. And we didn't have any questions, although there's one that just popped up. Let me just read this, Rich. So somebody says, all photos seem to be side views. Can you talk about best photo views for ID? I was trying to get head and back and rear and side. Do you want to talk a little bit about what aspects to photograph, if that's the way you're doing Bumblebee ID?
Let's hold that till the end, Leif. Let's try to get through this. We have some slides about that later, and maybe we can get to that later. And if we don't cover it here tonight, that is something we cover in detail on our Atlas sites and in our other trainings. So let's keep going with the ID if we could. Does that make sense?
Yep. Okay, so I'm going to talk about those that have yellow and black stripes, with T1, the first abdominal segment, being black. We'll start with the very common Vaznasenski Bumblebee. This is a bee found broadly along the west coast, though not especially far inland in most cases. So you can see from the range map that it occurs from Baja to extreme southern British Columbia, and almost all of the records are within a few hundred kilometers of the coast. However, the bee does occur in a wide range of habitats, including coastal at sea elevation, as well as up into the Sierras, the Cascades, the Blue Mountains. This bee is very common and seems to be adaptable to a variety of habitats. It is doing just fine, we think. So this is not one that we think is of conservation concern. It can be confused with Colognosis, which we'll talk about in a second, with Hunty Eye. The three of them together, we sometimes refer to as the yellow-faced bees. And then there are several others that do not have yellow face hair or have less of it. And we can also confuse them with this species also, that would be Fritzerson occidentalis. So Vaznasenski Bumblebee has a square cheek, just about as long as wide. It has short, even hair. I would say not exceedingly short, not as short as Crotchy Eye, but it's nice, even hair. The face and the vertex are bright yellow, sort of lemony yellow hair. There's no admixture or just a tiny bit of black hair on the very edges of it, but it should be all yellow. That front segment of the thorax, also all yellow. And then from there back, we have black hair on the thorax, black hair on the abdomen, except for T4, the fourth abdominal segment, which is yellow, as Rich is delineating there. And so I want to just point out this photo is a relatively nice one, but it is not possible to see all the different segments of the abdomen. So I'm referring back to that question we had. Here's one where no matter what your level of expertise, I don't think you could be confident of finding all of the gaps between the 6 segments. You can definitely see the ones that Rich has outlined there from 4, 5, and 6, but maybe not so much more anterior on the abdomen. So sometimes if you only have one photo, you are limited in what you can do as far as ID goes. And so, this be an important thing to distinguish it from Colognosis is that on the underside of the abdomen, which is called the sternum, and that's where this purple arrow is pointing upwards and to the left, there is all black hair. So we're not going to find any yellow hair underneath the abdomen. And so for most bumblebees, we're not concerned with the sternal part of the abdomen. We don't talk about the hair color there for most bumblebee species. For this one and its close relative, Colognosis, or the one that it closely resembles, Colognosis, this is an important characteristic. So we definitely want to pay attention to that. And if you think through this, if you are a photo-based bumblebee identifier, you are going to need a photo that shows those hairs really well. Like this photo, even if it did have yellow hair there, I don't think you could make it out very, very distinctively. So just one other thing about this bee, look at the range. This bee occurs, as I said, at sea level, also up in the mountains. And so normally when we identify bumblebees, we want to put on our blinders about where we saw it, what the temperature was, things that are external to the body of the bee. But you can cheat a little with certain things, and one of them is geography. So this species occurs in the Sierra Nevada. The next species, Colognosis, does not. And so if you are in the Sierras and you can't tell which one you have, you can be confident, or at least quite confident, that it's Vaznasenski Bumblebee and not Bombus Colognosis or obscure or fogbelt bumblebee. Go ahead to the next slide, Rich. And so here is that bee. This is Bombus Colognosis. We call it obscure bumblebee here, but it is also commonly referred to as the fogbelt bumblebee. And you can see why. It has a distribution that's even more closely associated with the Pacific coast. Another one that occurs as far as extreme southern British Columbia, and then down into just to Southern California. There are a couple of historic observations for the San Jacinto range and the San Bernardino range, but I don't think we'll find it there anymore if we ever did. So you will not, if you're in extreme Southern California, you are not going to have to think about Colognosis. It's going to be Vaznasenski instead. That's the kind of thing I was referring to with the geography. Now, this is a bee that also has a square cheek, perhaps just a little bit longer than Vaznasenski, but it is difficult to rely on that character for this pair of species. So just a slight bit longer or square. The hair is longer, it's shaggy, but it's not too long. It's, as you see, it's just a little bit shaggier than Vaznasenski. It has that same yellow hair on the face and the vertex, on the front of the thorax, all yellow, the same black hair color pattern across the body. The T4 is also yellow. However, there are two differences. So T4 is all yellow, but it has some black hairs medially. In most cases, these bees will have some black hair and sometimes as much as a little triangular shaped wedge in the middle of T4, which you can't see in this photo. So we would want to have another photo to look straight at the top of the abdomen to see those black hairs. And then secondly, and importantly, this bee has that yellow hair on the underside of the abdomen, usually on S. So these are numbered just like the T's. So S3 and 4, I think it is, which Rich has just circled, and you can see them in this nice image. So this is a very hard bee to distinguish from Vaznasenski, but with practice, we think you can start to do it. And let's move on to the next bee, Rich, which is, I believe, is going to be Bendikii. This is the third of the bees that we call yellow-faced bumblebees. They're somewhat closely related to each other in the same subgenus of bumblebees and very difficult to distinguish sometimes. And so that's why we call them this. This one is actually not so hard to distinguish from the other two. If you're new to bumblebees, it will look very similar, but the distinctive difference with this one is that the yellow stripe on the abdomen is on T3, not T4. So if you look at those color pattern diagrams, you can see that it's the third abdominal segment that has yellow hair, not the fourth. This is something that you don't normally notice if you're new to bumblebees, but once you start paying attention to them and you start being able to count those segments when you see a live bee foraging on flowers, or better yet, if you have one restrained in a vial, it's obvious that the hair is on T3 and not T4. So that's the big thing. This bee also has a relatively square cheek. It has longer hair than the preceding two species, a little shaggy. The head, all the hair on the face and the vertex is going to be yellow. Otherwise, it's going to look quite a bit like the others, with one exception, which is on this slide. It is extensively yellow in central Washington. So we're going to see animals that look more like the diagrams to the left side that Rich is circling in that part of the West. There, it's going to be a little bit tricky to distinguish it from some other species, but it's going to look quite different from the other yellow-faced bumblebees. So we can go ahead to the next bee. Okay, so here's another one in this color pattern group. It is a mimic, let's say, of the others in this group. It looks quite similar, but there's one glaring difference that you can see right away when you see this bee in the field, and that is the absence of yellow hair on the face and the absence of yellow hair on the vertex. There just isn't yellow hair there ever, in my experience, ever. So look carefully, but you are going to look for black hair on the head. You are also going to look at a nice long cheek. It's so long, in fact, that this bee is sometimes referred to as having a horse face. If you look straight on at the head, you will see that its mouth parts, the bottom part of its head, are actually quite long. And if you can compare this one to a Vaznasenski eye or some other more common species, you'll get it right away. This is a bee that otherwise is generally similar in its color pattern to the yellow-faced bees. So that's Vaznasenski, Colognosis, and Vandykii in California. So the image here is of a bee from California, I'm guessing, from working from memory here, but that color pattern is typical in California. This close resemblance to the yellow-faced bumblebees. In other parts of the range, in fact, most of the range, which goes coast to coast, it is almost entirely yellow. It has a black face, a black vertex, all yellow thorax, and then yellow on T1 through T4 and black on T5 and 6. As you'll hear about this again. But in California and parts of Southern Oregon, you're going to see the contact zone where they're somewhere in the middle of the two. But so we can move on to the next slide. And here is Bombus Franklin, Franklin's bumblebee. This species is, as you probably have heard, in deep trouble. It was added to the endangered species list last year. It's a species of greatest conservation need in Oregon and California. This bee has the smallest known global range of any bumblebee species on Earth. And that range is about 120 miles long and maybe 70 miles wide. And so that you can see the map here, that is everything. That is the entire range of this species, Southern Oregon and Northern California. So we're talking about Mount Ashland, the Marble Mountains Wilderness, the Klamath and Siskiyou Mountains. We have not seen this bee in 15 years, so it may be extinct. This is not for lack of looking. However, it does occur in pretty remote habitats at higher, mid, and upper elevations, and so it could be hiding out in one of these places. Indeed, the most recent observation is from a place where it had never been found before, and it was found by a master's student from Humboldt State who was working on a project and not expecting to find it. So I'm not trying to say that you could find it in a drastically different area of the West, like Montana, but I am saying that it's possible that it's hiding out somewhere in that range. Now, this is a species that's related to the Western bumblebee, if you've heard of that one, which is also of conservation concern. It's a relative of the rusty patch bumblebee from the Midwest and the eastern part of the continent. So most of the bees in this group are in trouble, and we can talk about that later if you're interested. Anyway, those bees, including this one, have a short cheek. And then with Franklin eye, we're going to see both black and yellow hair on the face and vertex. We're going to see a distinctive yellow band of hair on the front of the thorax, but it's going to be invaded by a sort of oval or horseshoe-shaped black notch on the thorax, on the center of the thorax. So the black hair that you might see between the wing bases is going to go into the front yellow band, as Rich has just nicely delineated there. And then we're going to see all black hair on the rest of the body, with the exception of some yellow hair down at T5 and sometimes T6. So this is a bee that will appear to be quite dark. You'll see that yellow stripe on the thorax, but you'll mostly see black hair. And you'll see just a bit of yellow hair or pale hair at the very tip of the tail. An extremely rare bee that we don't expect anyone's going to find, but we are looking, we are hoping to find it. So go ahead, Rich. I have a couple of things to tell you about this bee. If you do go looking for it, that's wonderful. And Xerces has a scientific collecting permit from the state of California to work on this bee, and from Oregon. However, until the bee is formally, until the, I think it's the recovery plan needs to be written by U.S. Fish and Wildlife Service, until that time, they don't want us to publicize the locations of new observations, should we make any. And this is just to protect the bee. We don't want a ton of people rushing out to trample the habitat before the biologists get to take a close look. So we hope you will find this bee or think you have found it. But if you do so, please contact someone from Xerces or a state fish and wildlife agency. to let us know about that before you post to social media, because we've sort of promised Fish and Wildlife that we'll tell people to do that until sometime this summer when we can start to be more public about this. And so go ahead, Rich. If you choose to use Bumblebee Watch, our data management platform for Bumblebee data, that's wonderful. We hope you will. But one way you can share photos with us without divulging the location of a potential Franklin's bumblebee population is on this screen that you see here. It's the part of Bumblebee Watch where you add photos of your bees, and there's a little spot. Thanks. It's step 2. There's a little spot where you can check for private, and that will just make the record visible to Rich and myself and other staff, but not to the general public, and that will protect the bee in the way that Fish and Wildlife has asked us to do. So go ahead. And now I'll turn it back to Rich to talk about bees with white hair.
Okay, thanks, Leif. Yeah, jumping right in here. First species we're going to talk about is Bombus Depositus. This is another, what I consider more of a sort of sagebrush steppe, lower elevation species. It's got shorter hairs. You can notice if you look at the picture on the right there that you can pretty clearly delineate the different tergal segments. This species has a very long cheek with a black band between the wings. Its hair on its face, and usually the hair in front of the wings is kind of a pale white color or a cream color. And behind the wings and the entire abdomen, with the exception of the 6th tergal segment, are kind of that burnt orange color that you see in the photograph here. There's no filters on this bee. That's really what it looks like in the wild. This is a large-bodied bee as well. The queens are exceptionally large, and the workers are quite large as well. Next species is the western bumblebee, Bombus occidentalis. Again, this is a species of conservation concern. It's been petitioned for endangered species protection. It's a species of greatest conservation need in Washington, Idaho, Oregon, and California. And this is you can see the decline in the species when you look at the distribution map. We have a really wide distribution historically, including at low elevations, all the way from the coast of Oregon, Washington, California, and British Columbia, all the way to the Rocky Mountains. And this species has more or less disappeared west of the Cascade Sierra Crest. There are exceptionally few records west of that Cascade Crest, with the exception of some areas on the Olympic Peninsula and on Vancouver Island up in Canada, where the species does seem to be extant. But in lower elevations, we don't find the species anymore in the Pacific Northwest, or for the most part in California, or at least haven't yet, hoping that the atlas turns this species up. This species has a short cheek. The females of the species have predominantly black face and black vertex, although sometimes you will see some yellow hairs mixed in there, as you see in this individual. The majority of the thorax is going to be yellow or black between the wings and backwards. The first 3 tergal segments are generally speaking black, and then the second tergal segment, or I'm sorry, the 4th and 5th tergal segments, and the 6th as well, sometimes have bright white hairs on them. You'll notice if you look at the color patterns, particularly for the worker, there are some individuals that don't have that snow white hair on them. And these are individuals we typically associate with sort of coastal California individuals, where that white hair fades to more of a cream color, kind of like we would see in Bombus Kensis. So something to be aware of if you're doing surveys, particularly in the western portion of California, is to keep an eye out for this species. And it may not have that bright white hair that we so often talk about. And it will often, or usually, have yellow hairs on T3 and sometimes also on T2. And the yellow hair on T3 is also possible in the further sort of east you go in Oregon, Washington, and Idaho as well, especially into the Rocky Mountain populations. In Idaho, almost always will have some yellow hair there on T3. I guess I also just want to remind you all that and this came up when we were talking about Vaznasenski Eye versus Colognosis. There was some confusion about yellow hairs on the underside of the abdomen. I want to remind you that right now, Leif and I are only talking about females. So the males can be quite different from this and quite variable. So right now, just as a reminder, we're still only talking about females, which are the most reliable to identify from color patterns. The next species we might find with pale or white hairs is Bombus Kensis. You'll remember earlier we talked about some of the other Pyrobombus, and particularly Bombus Flavifrons and Bombus Mixtus. We talked about the degree of sort of intermixed or cloudy hairs here on the thorax. And with Bombus Kensis, almost always, you'll just see a little bit of yellow here at the front of the thorax, and it just gets darker and darker the further back that you get on that thorax. And that is the character that we're really cueing in here, and you'll see it in the cartoons as well. We see cloudiness sort of in front of the wings and more dark the further back that you get. So that's a key character to cue in on. You'll also often find this notch in the middle of the second tergal segment, a black notch that we don't see on Bombus Flavifrons. And then again, these pale hairs that you see at the back of the abdomen here on the tergal segment 4 and 5. This species also tends to have really shaggy hairs. The hairs are long and uneven, as you can see along the thorax here. It doesn't look like that crew cut like we saw on Bombus Vaznasenski Eye. I'm just going to bring this slide up again. This is the same slide that Leif showed you with Bombus Franklin Eye. I'm including the species in ones with white hair as well, because the species can also have pale white hairs here. And because it's of conservation concern, we want to remind you of it as frequently as possible. I have nothing new to say here other than we hope you go out and try to find this bee. And I put some resources in the chat to hopefully help make that happen. And with that, I'm going to pass it back to Leif to talk about cuckoo bumblebees.
All right, thank you. Okay, let's get right into it. So we have just 3 cuckoo bumblebees to talk about in the West, in our area of the West, I should say. So the first is Bombus insularis, the indiscriminate cuckoo bumblebee. And I'll say just briefly a little bit about the life history here. These cuckoo bumblebees are the ones that are parasites on the other bumblebees. Instead of founding colonies and having a social structure with queens, workers, and eventually males, in this case, the female attacks a host colony of bumblebees and takes over. Eventually, the host colony produces her offspring and nothing else. So that is why we call it a cuckoo bumblebee. It's similar to those birds. They're very fascinating animals, and there's lots more to say about the life history, but we're going to leave it at that for now. So this is the most common, I'd say. Oh, it's among the pair that are most common in the West of the cuckoo bumblebees. This species uses many other species as hosts, so it's somewhat flexible in host choice around its range. And you can see it has quite a large range, going all the way from maritime Canada in the east to Alaska, although the heart of its range is really the montane areas of the West. So you can hopefully see that in the global range map. This is a common species, and so you're going to have to remember that you need to figure out how to identify cuckoo bumblebees, and our best guess, our best character to give you on that is the shape of the hind leg tarsus, where the pollen basket would be, but it is not in either the males or the females, right, of the cuckoo bumblebees. So if you can get to that, then we're going to talk about distinguishing the 3 species here. So insularis has a short cheek. That's somewhat typical of cuckoo bumblebees. It has kind of a thick head. It has there seems to be a bunch of area behind the eye, the back cheek area. This is actually referred to as the cheek in bee taxonomy. There's just a lot of head from the side view there. It has a distinct patch of yellow hair at the base of the antennae, which you can see in this image. And it also has yellow hair on the vertex. This is the only one of the 3 species of cuckoos we're going to talk about here that has this yellow patch of hair. It's distinctive, it's easy to spot, and once you learn it, it's pretty easy to stick with it here. So you're also going to be looking for a thorax with that's got yellow hair, but it's bisected over the top with a black band, a largely black abdomen, although we are going to get varying amounts of yellow hair on the sides of the abdominal segments, T3, 4, 5, and even 6. There is typically black hair centrally, medially on T4, I think that is. So let's move forward, Rich, and we can compare to the next species. So this is Bombus Flavidus, also known as fernald cuckoo bumblebee. And the species name, the Latin species name, was until recently Bombus fernaldi, but the species has been revised, and we now understand that this is part of a species that occurs around the globe in northern places. And so we have Flavidus in northern Canada and in montane continental United States, but also we see it in Siberia and other parts of Asia and Europe in northern and montane places. It's part of a variable group that is currently thought of as one species. This species is also common in the West. You should expect to find this. You're going to contrast this one to Insularis by the fact that the face hair is distinctively all black. And you can see that purple arrow pointing to the front of the head there, a nice black patch. This bee does have yellow hair on the top of the head, so on the vertex, but it has black hair between the eyes where the antennae originate. And that is a distinctive character to tell you that this is not Insularis. Also, we're going to look at, so the color pattern is somewhat similar. There's more yellow hair on the thorax than for Insularis. There's often just a spot, a large spot of black hair centrally, as in this image, as in this photograph, although it can be broader. Rarely we see this bee looking a lot like the yellow-faced bumblebees, the Vaznasenski and Colognosis, as you can see in those color pattern diagrams on the right. But typically it's more like the one in the photo and the images farther to the left. There's a purple arrow pointing to the tip of the tail that I want to explain. This is not super easy for you to see, but where Rich has got the cursor there, the very last segment, the T6, which is also on top of the S6, the S6 has these special little flaring projections that come out from the side, but they're not particularly big. If you were to look from the top of the abdomen downwards, you would not be able to see the sternal segment underneath. This sounds kind of esoteric, but we'll explain it better on the next slide. So to compare this species to Sucklii, which is the really rare one, we want to look for an absence of these flangy parts of the body. It's actually the exoskeleton of the body sticking out sideways under the top of the abdomen. So Rich, why don't you go forward and we can talk about that. So this is the last of the three cuckoo bumblebees. It's cuckoo bumblebee or Bombus Sucklii. Let's just start with that sternal segment S6 character. So look in the bottom right-hand part of the photos, and you'll see this is looking downwards from the top of the abdomen, not under the underside. And the flangy parts that are sticking out, that's actually the sternal segment. So it's for whatever reason, it's very broad, and you can distinctively just make it out looking at the top of the abdomen. That's a good characteristic for this bee. You'll also be looking for black hair on the face, on the vertex, the lack of, obviously, of any pollen carrying apparatus, and then it does have yellow distally on the abdomen. So starting on T3, but especially T4, we see some yellow hair. And importantly, this bee has yellow hair on the sides of the thorax. And this doesn't actually distinguish this bee from the other two in our area, but it does distinguish it from another species that's found farther north into Canada and Alaska. So that yellow hair on the side of the thorax under the base of the wing is distinctive for almost all populations of Sucklii bumblebee, cuckoo bumblebee. This is a very rare species. It's been petitioned for listing under the Federal Endangered Species Act. It's a species of greatest conservation need in California, Washington, Oregon, and Idaho. It is very, very rare in the continental US. So please look for it and please let us know if you see it. With that, we can move on. And I will turn it back to Rich.
Okay. So that's it. We have now introduced you to all of the species that you would encounter in the Pacific Northwest and California, and probably most of the rest of the West as well. We're now just gonna we don't have a whole lot of time left, so wanted to zoom through some of the lookalike groups. We've already talked about this, so I don't think we need to spend a whole lot of time on it, but just wanted to put some of these lookalikes on the same slide so that we can start talking about them and talk you through how we might be able to differentiate some of these species. So the first group here, the yellow-faced, yellow-striped bees. This is Bombus Colognosis, Bombus Vandyckii, and Bombus Vaznasenski. The fog-belt bumblebee, Vandyckii bumblebee, and yellow-faced bumblebee from left to right here. And again, just as a reminder, we think about Bombus Colognosis and Bombus Vandyckii as having longer and shaggier hair. And you can see that if you look sort of above the thorax here, you can see hairs sticking up. We don't see any of that on Vaznasenski, right? Same thing with Vandyckii. You see little hairs sticking up and longer hairs, especially here on the abdomen. Likewise, on sternal segments 3 and 4 on Colognosis, we see those yellow hairs on the underside, and we see the same thing actually on, I think, 3 through 5 in Vandyckii, so longer yellow hairs, and the underside of Vaznasenski, as you can see here, are black. So those are the things that we're looking at to differentiate those species. And then again, the reminder that this is tergal segment 3 here for Vandyckii. And you can see this is somewhat common here to have actually a little bit of black at the front of that third tergal segment as well. So just again, looking at the thorax there, the difference in hair length, even on Vaznasenski to the far right, and less even on the other ones. And then the location of the yellow segment delineated from those numbers. Closer look at those yellow hairs, very clear on Colognosis on the left, Vandyckii in the middle, you can see the yellow hairs, and then pointing to the black hairs in Bombus Vaznasenski on the far right. And then again, here is another close look at those yellow segments on the sternal segments. So here is Colognosis. You can clearly see the yellow hairs. Here is Vandyckii. Again, you can see it on 3 segments on the underside of the abdomen. And then all of these hairs that we can see sticking up here on females of these species, again, this is just females, are going to have black hairs in Vaznasenski and yellow in the others. Male Vaznasenski, and this is very confusing, have a lot of yellow hairs, just like these other ones. So that makes male identification much trickier. The next group I wanted to talk about are what I'm calling the cloudy pyrobombus. Bombus mixtus on the left, Bombus Flavifrons in the middle. I'm sorry, Bombus Kensis on the left. See, I even get confused. Bombus Flavifrons in the middle and Bombus mixtus here on the right. And what I want you to pay attention to first is, remember, with Bombus Kensis, a little bit of yellow in the front and just gets darker and darker as we move back. With Bombus Flavifrons, we see yellow in the front and still some yellow behind the wings here, right? So an equal amount in front of and behind, much different than in Kensis. And then mixtus usually has fairly bright hairs on behind the wings. So good differentiation there for these 3 species that otherwise can be quite confused. And then also here on Kensis, remember we had that break at the back of T2. Sometimes in Flabberfrons, we see a break at the front of T2. And then remember, Leif mentioned that crescent of yellow that we sometimes see on Bombus mixtus. Next 2 I wanted to quickly talk about is Navidensis versus Morrisoni. Remember, Navidensis was a long-cheeked bee. Morrisoni on the right here is a short-cheeked bee. Navidensis on the left has a black vertex on the top of its head. Morrisoni on the right has a yellow vertex, so yellow hairs on the top of its head. And again, we can see this in the photograph here. We can see a black vertex and a spot on Navidensis, and a yellow vertex and no spot on Morrisoni. And then again, see that little crescent of yellow also on Morrisoni down on T4 there. Centralis versus Flabberfrons. This is going to be another very tricky group. Mainly, we're going to cue in on the thorax here. Cloudy hairs on Flabberfrons to the left, no cloudy hairs on Centralis to the right. The degree of cloudiness on Flabberfrons does change throughout its range, but largely you're going to see a much more admixture of black hairs into those yellow hairs on Bombus Flavifrons versus Centralis. We can also use cheek length for this. Flavifrons does have a bit of a longer cheek than Centralis. Appropriate to note here that the size of the individual is going to impact the length of the cheek. So we really want to be paying attention to the ratio, right? It's the width to the length, not just the length here. So I put this picture here so you could see the difference in cheek length, but it's not the whole picture. Remember, we're actually looking at the width, so we need to pay attention to how wide the mandibular hinges are to the length, not just how long it is. So this is not quite a fair comparison, but it is something I wanted to show. The last, I think, species group is this Melanopidges versus Edwardsi. So here, these two, the black color form of each of them. With Bifarius, we're going to see a black notch behind the wings here, and we don't see that black notch, or it doesn't extend all the way to the back of the thorax in Edwardsi or Melanopidges here on the right. And then with the back of the abdomen, we don't see any yellow on T5 in Bifarius, but we do see yellow on T5 in Melanopidges. So that's the other thing we're going to cue in on. Oh, and then we're also going to talk about the yellow, red, yellow, reds, the other close lookalike group. So Bombus Rufa Sinctus, remember, we're going to key in on that crescent of yellow on T2. That's a key factor for Bombus Rufa Sinctus. For Bombus Silvicola, we're looking at very long hair, long shaggy hairs, and then yellow all the way down to T5. So that's another cue in that we're going to look at for Silvicola. Bombus Bifarius, we're going to look at that black notch behind the wings and usually some black on T2. Bombus Hunty Eye is our sort of never varies, evenly cut, kind of like Bombus Vaznasenski. The hairs are even length. It always looks very similar and is the most sort of even cut of all of these species. And then finally with Bombus Melanopidges, we have those cloudy hairs at the front of the thorax there. And T4 is also cloudy, a mix of yellow and black hairs on T4 and the red color form for the species. Finally, some Bombus Franklin and a couple that you might confuse this with. So this is Bombus Vaznasenski here on the left, actually. Notice how much more extensive the yellow hairs are on Bombus Franklin over here, creating that sort of horseshoe shape. We don't see that in Vaznasenski. Even if it's just a bit of a crescent here, it doesn't extend all the way to the back of the wings as we would see here in Bombus Franklin. So that's something to look in. Also the yellow face here versus the black face on Bombus Franklin. Also comparing Bombus occidentalis to Bombus Franklin. Again, we're looking at that crescent horseshoe shape that we see here in Bombus Franklin. And we don't see that in occidentalis. And the yellow or the white hairs rather that we would see on occidentalis are going to be more extensive, extending all the way across T4 and T5, whereas that would be much more limited in Bombus Franklin. Leif, moving quickly here, I'm going to pass the males back over to you.
Thank you. All right, thanks. Yeah, we're running out of time, so I'm going to run through about 5 species pairs of male bumblebees to talk about identification of males. They are less than 50% of the bees you'll see, but of course they are common and necessary to the propagation of more bumblebees. And so they commonly turn up. And if we often just teach the female side. And so one of the challenges for beginning naturalists who are interested in bumblebees is the presence of all these males. The males can look very, very similar to each other in some species pairs. And so we'll get into some of that. In standard taxonomy in using identification manuals for identifying male bumblebees, you will often have to use characters that are morphological in nature and are beyond the scope of what we do in our bumblebee atlases, namely pulling out the genitalia of the animal when it's dead and before it has dried and become brittle. And so that's what these pictures are on the right. Those are the genitalia of bumblebees. And you can see the names. Those should be some familiar names there. We have penis valves and a gonostylus and a bunch of other parts here. These are the parts of the male bee that fit into the female. They're distinctively different in each species. And so sometimes when we have no idea the distinction between one color pattern and another, we would have to look at the genitalia to really know for sure. But I'm going to talk about color pattern for those where we can distinguish them here. So go ahead, Rich, to the next slide. And we'll compare males of Flabberfrons and Van Dyckii. And now these are not the only possible color patterns for these two species, but they both commonly occur in largely yellow forms. So on the left, we see Flabberfrons. He's got abundant yellow hair on his face and vertex and thorax, and most of the abdominal segments are yellow. But the arrow is pointing to I think it's 4, that has some black hair mixed in with the yellow. And you can see that on adjacent segments also. On the right, we're looking at Van Dyckii. Remember, this is one of those yellow-faced bumblebees that all resemble Vaznasenski Bumblebee. Well, in the case of male Van Dyckii, they are almost entirely yellow like this. They don't have extensive black hair, although they do have some black hair across the abdomen. But I want you to notice that where the arrow is pointing and where Rich is indicating, that black color is the integument of the bee. It is not the hair. And if you look closely at the actual hairs that you can see in this lovely photo, they are all yellow. So this is one way to distinguish those nearly all yellow Flabberfrons from the standard look of a Van Dyckii male. I will say that in that area of, I think it's Central Washington that Rich mentioned earlier, Van Dyckii females look different and the males can look quite distinctively different also. You can go ahead and we will stick with Flabberfrons here. So in other areas, well, in other populations, let's say, Flabberfrons males often have red hair on the abdomen, similar to those females that also do. And so on the left here, this is an image of a male Bombus Flavifrons. Remember that this is a guy that has lots of mixed black and yellow hair on the thorax. We said that for the female, and that's true for the male also. And so that arrow is pointing to a good mix of black and yellow hair on the distal part of the thorax. And then here we are seeing 2 red stripes of hair on the abdomen. And the arrow is meant to be directing your attention to the presence of a couple of black hairs, which are pretty hard to see in this slide, but they are there in that image if you zoom in on it. And so even when Flabberfrons has red hair stripes on the abdomen, we are typically going to find some degree of black hair on T4. I think that's T4. By contrast, Centralis on the right has the same gross overall color pattern with a mostly yellow body with some black hair in the center of the thorax and 2 red stripes at T3 and T4 in the abdomen. But notice this bee is all yellow where it's yellow. It does not have a mix of black hair with the yellow hair. Maybe you'd find a stray black hair or 2 here or there, but it is very brightly yellow on the thorax where we have yellow hair. And then on the abdomen, we're going to see a lack of black hair mixed into that red. And that is one way that you'll distinguish Centralis from Flabberfrons. The other thing, we've talked about cheek length a bunch for the females. Flabberfrons males can be expected to have a relatively longer cheek length than other male bumblebees, not all of them, but many of them. However, it's a little bit different with the males. Sometimes the distinction of the cheek length is not quite so obvious as with the females. And just to recall that for Centralis, we told you that for the females, the cheek is just a little bit longer than wide, but not as long as in Flabberfrons. Go ahead to the next one, please. Now we're going to compare Insularis, which was the indiscriminate cuckoo bumblebee, to Flavidus, which is, or Fernaldi, which is one of the other cuckoo bumblebees. So first of all, you will have figured out that it's a cuckoo, and then you're not sure which one, and it's male, and oh no, they all look alike. So on the left, we see Insularis. This guy is going to have yellow facial hair like the female, but that's not necessarily absent from Flavidus in all cases. But what you will see in Insularis that's distinctive in this mostly yellow body in these populations is that on T4, we have black hair right in the center of the yellow band. So the yellow band is broken by black hair. And that's just a distinctive thing for this species that you generally will not see in Flavitus or in Suckly Bumblebee. And so on the right, we're looking at Flavitus, also known as Fernaldi. And you can see an all yellow, more bushy sort of stripe on T4. In addition, in almost every male Flavitus that I've ever seen, no matter where on the continent, it has red hair on T7. So remember that males have 7, not 6 abdominal segments. In almost every case, Flavitus will have reddish hair at the tip of the tail, and that should help you. If you know it's a cuckoo bumblebee, you've decided it's a male, and you see this red hair, it's going to be Flavitus, and not Insularis in this comparison. So go ahead. All right, so here we'll compare Melanopagus with Silvicola, and on the left, I'm showing you Melanopagus, and this is one of the red-banded individuals. This is not like Edwards Eye in California, the form that used to be called Edwards Eye, where it's the red would be replaced by black. Remember that in the females, we had a good admixture of yellow and black hair in the front band of the thorax, and we see that here in the male as well. By contrast, Silvicola, that high elevation, moderate to high elevation species, has an all yellow band of hair at the front of the thorax. And in general, we think Silvicola tends to have paler yellow hair. I don't know if we can depend on this, but this pair, you can definitely see the difference in the hue of the yellow. Go ahead and advance to the next slide. And here we'll compare Melanopagus and Bifarius. We'll wrap this up very shortly. I understand we're a few minutes past 7 p.m. here. So on the left, we're seeing the Edwards Eye form of Melanopagus. And on the right, we're seeing Bifarius, the way it looks in California and some other parts of the West, that is lacking red hair. And so I'm once again pointing your attention to the back side of the thorax, the distal end of the thorax that kind of faces the abdomen. And remember that in the females of Bifarius, there's a black notch of hair. Well, we see it in the males also. So it's a little bit bushier in its hair, but you will definitely see black hair going all the way through that back hairband on the thorax. And with Melanopagus, it's going to be a solid yellow band. So go ahead to the next slide, Rich. And unfortunately, I'd like to be able to show you how to distinguish Colognosis from Vaznasenski, but as we've already learned, that is not possible in males unless we pull out their genitalia on pinned dead specimens, and we can see some differences. And I won't get into what those differences are right now, but I think you can see if you squint some differences in the shape and the morphology of these various body parts. And so I think that we're going to have to leave it there. And let's see, Rich, do you want to take any last questions?
Well, maybe we should just quickly handle this. What pictures should I take? Side of things. Sure. And there's also a question about if I'm using iNaturalist, do I also need to use Bumblebee Watch? And I will say that if you are a member of the Pacific Northwest Bumblebee Atlas or the California Bumblebee Atlas, and you are conducting a survey, so largely the focus of these projects are to conduct standardized, effort-based, timed surveys in equal areas. You'll need to submit those data on Bumblebee Watch using the data sheets that we have there. If you are submitting incidental sightings, so you've just found a bumblebee and you'd like to have it identified, iNaturalist is a resource that you can use. I will say that the identifications on iNaturalist are crowdsourced. It could be that Leif and I look at them. It could be that that somebody else identifies them and makes that a research grade observation on iNaturalist. And there are a lot of very good research grade observations on iNaturalist, and I don't want to say that it's not a good resource, but there are also some misidentified research grade observations in iNaturalist. If you submit it to Bumblebee Watch, it means that it's very likely that it's Leif or I that will be looking at those photographs. And I don't want to pretend for 1 second that means that there won't be misidentifications, because this is an imperfect science, and we will get identifications wrong. And we hope that when we do, or you disagree with us, that you will challenge us on this and have us relook at the photos and let us know why you think it's some other species. We welcome those conversations, but they are all expert verified on Bumblebee Watch and not community sourced by anybody. So that's the difference. And you're welcome to, for those incidental observations, you're welcome to use whatever you find easiest to use. As far as what photographs to see or what photographs to take, especially if you're chilling bumblebees and taking detailed photographs, generally we need to see the hairs on the face and the vertex. All these features we've been talking about for almost every taxa, right? We want to see, ideally, have some notion of how long is the cheek. We want to see the hairs on the face and the vertex. Ideally, we want to see the hairs on the thorax, both above the wings and between the wings, but also sometimes below the wings. There are characters that we look at there. And then ideally on each segment of the abdomen, including sometimes bumblebees will fold their wings over their abdomens. And sometimes we want to pry those wings apart so that we can make sure to see the center of T2, which was often a crescent that we see. And the reason that most of the photographs that you see are like the photo that we see here from the side is because I can see a lot of those features here, right? I can get some notion of cheek length. I can see the hairs on the face, and the head is yellow, and I can see below the wings and above the wings, and I can see most segments on the abdomen. And in particular on this one, I can see, that there's maybe a tiny hint of red there on T7, letting me know this is, a male Bombus Flavidus. So that's it. Like sometimes you don't have, that doesn't, I think I probably mentioned like 7 characters that I need to see to identify a bumblebee. That doesn't mean 7 photos. It might mean 1 photo or 2 photos or 3 photos. And generally on Bumblebee Watch, we allow 5 photos, but usually you can get all those characters with just a couple. Both of our atlas sites, California Bumblebee Atlas and Pacific Northwest Bumblebee Atlas, both have recommendations for photos, as well as how to work a camera. And the other thing I will say is both of our groups, or both of our Atlas projects, also have private Facebook groups, and those are good places for resources about photos and how to take good photos as well. So I would encourage you to reach out to other volunteers and learn what they're doing and how they're doing it. Because in some ways, their experience is more appropriate than mine, just because I've been doing it longer and have thousands of these under my belt, as opposed to just getting started. So if you're just getting started and want some help, I would encourage you to use those Facebook groups. Leif, anything to add there?
Yes, I'll just answer this question from James quickly. It came up while you were speaking. He asks, after a survey, how should we report no bumblebees seen? And so if you are a participant in one of the atlases, the PNW or California, or maybe one of the others, that data, the fact that you did not find bumblebees is very valuable to us. So we want you to submit that survey data. And so you'll go to Bumblebee Watch, you will behave as normal, you'll enter the survey information, and then the second step is to enter the bees themselves. You will just skip that second step. If you want to, you can leave us a little note in one of the notes fields saying, I saw no bumblebees, but we will understand that when we look at the survey data. If there are no bees associated with that survey, we'll get that it's a 0. So please do submit those data if you're a member of a Bumblebee Atlas. We really value that information. And I think with that, we will wrap it up. So thank you very much, everybody, for joining us. And I hope that it has been productive for you, and we hope to see you next time. Oh, Rich, you're on mute.
Had to do it once. It's a requirement for every call that you're on. You got to talk while you're muted at least once. Just as a reminder, we did record this. We will download it and shouldn't need a whole lot of processing. We'll just get it posted to the website. We'll post it both at California and PNW Bumblebee Atlas sites, and also to the Xerces YouTube channel. So if you want to watch it again, you're welcome to do that or share it with friends or whatever. So yeah, hope this was helpful for everybody, and we will maybe do it again sometime. Maybe start with the males and work our way to the females or something like that, so we can do it again. And we had some other things we wanted to get to, but didn't. That would be fun. But maybe we'll do that on social media or something and find a way to do it and have some fun with it. So yeah, thanks everybody. Appreciate your time and efforts.
Have a good night.
And just as a reminder, at least in the Pacific Northwest, survey season begins June 1st. So hopefully this was a good primer for survey season to get out there and start your surveys. Super excited for the season to launch. And we also have a full Pacific Northwest Bumblebee Atlas training on June 4th, as well as some in-person field events. So we're running a field event in Vancouver, Washington, just outside of Portland, from 1 to 4 p.m. on June 4th. I'm going to be in the field, helping folks learn how to ID and catch bumblebees. And my colleague Julie Combs will be in Seattle, leading a similar workshop. So if you're interested in those, check out our events page on the PNW Bumblebee Atlas page, and feel free to register. We'd love to see you out in the field and at our webinars. So anyway, yeah, again, thanks a lot and have a great evening.